Protein Databank. It's basically a repository for all the macromolecular structures and particularly proteins that have been determined. As you can see, there's over almost 160,000 different structures within here, and it's really quite easy to navigate. If you want to find a protein, all you really need to do is type it a search up here. For example, let's see if we can find catalase, and brings up very quickly a whole list of different protein structures, all with of catalase. And as you can see, there's many of them from different species, and crystallized and determined in different ways. So let's just take a peek at one. If we look at here, then what we see is a link to the paper where the description of the structure was made. And further down here, you'll get all sorts of sequence information. Now, this is a particularly important one. This shows you what other molecules were co-crystallized, or what other molecules were associated with the structure. So in this case, we've got a sort of porphyrin. Now, if you want to take a quick peek at the molecule, one of the easiest ways to do that is to just look at the structure. Click there. And you get this very nice 3D representation of the structure, and you can use your left mouse button to move it around, and you can right mouse button to move it up and down, and scroll button to zoom in and out. Okay, so this is a really nice way of looking at the structure very quickly, and you can manipulate it over here. So for example, you might be able to change it to a surface model, so that's the surface of the protein in great resolution. That one's probably a bit better. We can change the color scheme to secondary structures. So there you go. Now you can see all the beta sheets and alpha helices and so on, and you can change it to various other bits as well. So now we can see those heme groups in there. Okay, so that's a really nice, easy way of looking at the structures. But what I need you to do is use something a bit more sophisticated than that to work on structures. And to do that, we need a bit of software called PyMol. Okay, and I'll show you that now.